Thank you for your kind introduction. My name is Zhongliang Guo from the Aichi Cancer Center Research Institute in Japan. The title of my research is a multimodal framework combining sequence and topological feature for accurate protein-protein binding affinity prediction. So at first, let me give a short introduction about protein-protein interaction. So protein-protein interaction are essential for various biological processes, such as signal transduction, cell metabolism, and the immune system. Here we give some examples for protein-protein interactions, like the various antibody interactions, the cell and the cancer cell interactions, and enzyme and substrate interactions. So understanding how proteins interact with each other can help research uncover the mechanism of life and identify the cause of disease and design new drugs. So quantify the protein-protein binding, protein-protein interaction is not a trivial task. So the common metric for protein-protein interaction is protein-protein binding affinity, which can be measured by the equilibrium dissociation constant Kd and the Gibbs free energy delta G. So there are some experimental methods for protein-protein binding affinity, such as SPR, QCM, ITC, but all the methods are quite costly and time-consuming. So to overcome the limitation of the experimental method, various computational methods have been developed, such as molecular dynamic simulation, empirical energy function, machine learning methods. So compared with the previous two methods, machine learning method have the benefit of high throughput, and the accuracy of machine learning method depend on the model and the data set. And recent models have shown quite high accuracy. So this is a slide for the machine learning models. There are various machine learning models can be used for the protein-protein interaction prediction, such as linear regression, gradient boosting, and neural network. For all of the machine learning models, the feature is very important for the final accuracy. So it is so how to design an efficient feature extraction method is very important for this kind of for machine learning. So for proteins, there are two kinds of representations. One is the amino acid sequence, and the second one is the 3D structures. So based on the representation used by the model, protein-protein binding affinity prediction model can be divided to the sequence model and the structure model. So this slide, I show the sequence model. So for the sequence model, which are use amino acid sequence to predict the binding affinity. So the key challenge is how to encode the sequence information that for the binding affinity prediction. So many methods have been developed. So one of the most recent and promising one is ESM2. ESM2 is used in ESM4, which is a protein structure prediction model. And ESM2 is trained with hundreds of million protein sequence, and use it, the ESM4 skips the time-consuming step of multiple sequence alignment. So we also use the ESM2 to extract the sequence feature for binding affinity prediction. For the structure-based method, the feature extraction is also very important. And as we know the structure of the protein, so we can focus on the binding interface, and we can extract the distance pattern of the atom on the binding interface, and we can also extract the topological feature for the prediction. So the objective of our research is to develop a protein binding affinity prediction method which can have a high accuracy for which can be applied in protein design and other downstream tasks. So we consider three criteria for this task. First, the model should be high accurate, as the subsequent experiments are quite time-consuming. And the second one is that the model should be high throughput to handle the virtual screening and other large-scale calculation. The third one is that we want to apply this model to unseen protein complex. So we test the ability of this model, use the Lee one struct out cross-validation, and we will explain this in more detail later. So this is the structure of our model. The multimodal framework for protein-protein binding affinity prediction have 3 components. One is the Ceramid's ESM network, and this is for generate the sequence feature, and convolutional neural network is used to generate the sequence feature from the persistence diagram. And at last, gradient boosting tree is used to predict the delta G. Delta G is the change of delta G between the wild type and mutant type protein complex. So in our research, the benchmark dataset include delta G as instead of G, so we use delta G as a prediction target. So this is a sequence feature generator. We use the Ceramid's ESM network. So ESM2 is a pre-trained protein sequence and binding generator. It takes one chain sequence from the protein complex and generates the chain feature. It can only take one. So the protein complex feature is generated by takes the element-wise product of the 2 chain feature, and we generate the complex feature. So the structure feature generation can be divided into 4 steps. So in the dataset, only the wild type structure is given. So at first we predict the mutant structure from the wild type, and then we create the point cloud of atom on the interaction interface, and then we generate a topological feature of the atom at the interaction phase. So topological persistence diagram. So persistence diagram is a topological data analysis method that can capture the feature of connection and shape of the point cloud. And at last, we use the convolutional neural network to extract the structure feature from the persistence diagram. So the binding affinity is predicted by the gradient boosting tree. Gradient boosting tree is a well-used machine learning model. It can always achieve high accuracy with minimum hyperparameter tuning. So we use the gradient boosting tree to take the sequence feature and structure feature as input and to predict the delta G. So this slide shows the dataset we used to assess our model, to test the performance of our model. We use AB band and SCAMP dataset to test our model. The AB band dataset is an antibody binding mutation dataset. It includes over 1,000 data points and 22 structures. We focus on the 645 single point mutations. And the AB band dataset is quite homogeneous. It only contains the antibody and antigen complex. SCAMP dataset is an assembled dataset. It includes the antibody antigen and protein and inhibitor. So it includes over 3,000 data points and 148 structures. So we focus on the 1,131 non-redundant single point mutations, denoted as S1131. So SCAMP dataset is quite heterogeneous because it includes the antibody antigen dataset and the protein inhibitor dataset and other kind of complex. So the dataset is like this. The wild type protein structure is given by the PDB, and the mutation is given here. So we can know the wild type sequence and the mutant sequence, and the delta G is given here. Also, the dataset includes the temperature, pH, and other experimental method, but we don't use in our method. So we evaluate our method, use cross-validation and leave one structure out cross-validation. So cross-validation is a wild used validation method for machine learning model. So here is an example of a small data set with 3 protein complex structure and 9 record of the delta T. So in the regular cross-validation, we random choose 3 of the data from the delta T from the data set and train the model on the remaining 6 data and test the performance on these 3. As all of the protein is included in the training data set, so the model can learn the protein structure and sequence during the training step. And in the leave-one structure-out cross-validation test setting, the binding affinity data of 1 protein complex is excluded from the training data set. So the model is trained on the other 2 protein complex and tested on the held-out protein complex. So this method mimics the realistic situation where we have a new protein complex with no binding affinity data available. And so we can apply a trained model to predict the binding affinity change for this new protein complex. So this shows the result of our method. So we check the performance of our multimodal model on S645 and S1131 dataset, use tenfold validation and leave-one struct-out cross-validation. On the S645 dataset, the AB band dataset. It is the AB band dataset. Our model shows significant higher performance than the previous models. Here, our model is the highest. And in the leave-one struct-out cross-validation, which is more difficult task, our model still performs quite well. So on S1131 dataset, the SCAMPy dataset, this is a SCAMPy dataset, which is a heterogeneous dataset. The performance of our model is similar to the state-of-the-art model. But in the leave-one struct-out cross-validation, we consider that all of the models have low performance. So considering that SCAMPy dataset is a heterogeneous dataset which contains different kind of protein complex, this suggests that the predicting binding affinity across different protein families and interaction types is a quite difficult problem, as we only have limited data here. But consider the downstream tasks such as the protein design. So model that can work well in homogeneous data is very useful. So we consider our model as a powerful tool for the protein design and other downstream tasks. So at last, we conduct an ablation study to investigate the contribution of each component to the overall system. So we separate our multimodal model to sequence-only model and structure-only model. So the result is quite interesting. It shows that the contribution of the 2 features depend on the dataset. So on the S645 dataset, the structure-based structure-only model performs quite well, but in the S1131 dataset, we find that the sequence feature had a similar performance to that of the multimodal model. So as discussed before, the SCAMPy dataset contains a wide variety of protein complex. So the sequence feature generated by ESM2, and ESM2 is a pre-trained model on hundreds of millions of protein sequence. So it can capture a general protein representation from the protein sequence. So we consider that in the SCAMPy dataset, the sequence feature works well to capture the general feature. And in S645 dataset, so the structure feature works well. It is because that the structure feature is a homogeneous data set, and the structure feature can capture the useful information from the structure, but it cannot be generalized to the different protein families. And using both of the model can improve the performance and makes the model more robust to different data set. This is likely due to the gradient boosting tree can automatically select the feature from the feature pool that are effectively in predicting the target property. So this is the conclusion. So we developed a novel multimodal framework for protein-protein binding affinity prediction. This model achieved high accuracy, and we implement our model in Python and R. It is quite a high throughput and available on computer cluster. So this feature makes the model quite useful for virtual screening and other downstream tasks like the protein design. So we plan to conduct some further analysis on the model to invest what kinds of protein is difficult for the model in the one structure out cross validation, and what kind of mutation is difficult for this model to prediction. And we also plan to check the prediction accuracy on the multiple mutations. Thank you for your attention.
Thank you. Now it's open to discussions and questions from the audience. No? Oh, okay, please.
Hi, great talk. I think the ablation study results are very interesting because you show that some protein, they are more important for the sequence features are more important. In some, the structure features are more important. And your multimodal model is robust. They can choose between these two. But do you know, can your model show that which features they are used? Is that biased toward using more sequence feature in some cases or the structure features in some cases?
So the importance of the feature can be calculated because we use the gradient boosting tree.
Yes.
So the gradient boosting tree, we can calculate the feature importance. Yes, we can do that. But in our ablation study, we focus that for different data set, for the AB band data set, the structure feature can achieve a similar performance with the multimodal model. So it seems that so we think that on the AB1 data set, the structure feature is more important than the sequence feature. But to quantify the importance, maybe we should check the importance given by the gradient boosting tree.
Thank you.
Yeah, nice talk. So I want to know how many data points you used for developing the model. How many data you used?
So for the AB band data set, we use 645 data points. All of them are single mutation. And for the SCAMP data set, we use 1,131 data points. And also, all of them are single mutation.
So some of them would be structure, like all of them don't have structures, like all proteins.
Sorry.
So how many proteins they have structures in the data?
So can I show my slide back? Can you show my slide? So in the AB band data set, we have 32 structures. They are wild type structures. And so for the experiment, the testing, we just predicts the mutant structure based on the wild type structures. And on SCAMP datasets, there is over 100 structures, and all of them are wild type, and we predict the mutant structures based on the wild type.
So how did you predict the mutant structures from?
We use the traditional prediction, protein prediction methods, like the prediction modeling. Use
Like, did you use Foldex or other methods?
yes, these kind of things.
Yeah, yeah. And
Well, the time is up, so please, next question, very quick.
Just a very quick question. So this is like used for predicting the effect of the variance or just for the binding affinity between two protein sequences? So can we like get the variant effect, or it helps us in predicting only the binding affinity between two protein sequences?
So you.
So like, because you have shown here like the mutations that like this residue is getting mutated from D to A or something. So we get to know the effect of the variant, like the mutations
Yes.
in the protein structure, or we just get to know only the binding affinity?
Yes, but the variance maybe can be multiple mutations like this. So in our experiment, we will only do the simple mutations, and we plan to conduct some multiple mutations. That,
So only the binding affinity?
yes, only the binding affinity.
Okay, so like, have you addressed also the water molecules also?
Molecules, like the molecule and protein interactions.
Yeah.
So there have some other research about this, I think.
Okay. Thank you.